Hi there and welcome to PhD at Living. Today we're going to talk about how the human eye sees colors. I know that sounds like a lot of biology, but I promise the chemistry is going to be hot and heavy. With that, let's take a look at the electromagnetic spectrum. The electromagnetic spectrum, shown here in typically poorly drawn PhD at Living fashion, displays the various wavelengths and frequencies of electromagnetic radiation. What's electromagnetic radiation, you ask? Well, it's basically any form of light that carries radiant energy. Light can act as either a wave or a particle, which goes to the particle-wave duality of light that we are definitely not going to talk about today. So, for the purpose of our discussion, we're going to think of light as waves. I've thrown a number of things up here on the spectrum to act as benchmarks. We can see things like AM and FM radio waves have wavelengths on the single to tens to hundreds of meters scale. When I say wavelength, I'm talking about the distance between 2 points in a sinusoidal wave that have the same phase. So we're talking about peak to peak, trough to trough, or any point in the middle. As we continue down the spectrum, we get to microwaves, which have wavelengths on the centimeter scale, near infrared or IR waves at the micron scale, X-rays at 100 single nanometer range, and down to gamma rays at 0.1 angstroms or 10 picometers. Yes, I know you heard me say gamma rays, but don't think you're going to be Bruce Banner just because you're exposed to them. In fact, you'll probably just get cancer, so sorry to burst your bubble on that one. The electromagnetic spectrum, which theoretically covers wavelengths from the size of the universe down to about the Planck length, please don't ask. Fine, 1.6162293 times 10 to the negative 35 meters. Are you happy now? Is practically about 20 orders of magnitude, all the way from 10 to the 8 meters down to 1 picometer. Squeezed in here at about 10 to the negative 7 meters is a range of wavelengths from 400 to 700 nanometers. These 300 nanometers account for all the colors that normal humans can see. And by normal, all I'm saying is a human that can see the same range of wavelengths as a majority of other humans can see. Some people see more wavelengths, some people see fewer wavelengths. No intent to offend anybody on anything. Moving on. Let me hit that one again for you in case you missed it. In a roughly unlimited number of wavelengths, humans can see 300 of them. 300 out of infinity. Seems kind of small, no? Great, cool, awesome. So how do we change incident light into visible colors? Well, now comes the biology of the chat, which is unavoidable, but hey, sometimes biology is cool and interesting and barely distinguishable from chemistry, so let's get after it. Listen, the eye is such an incredibly complex and wonderful system that me pretending to know what's going on is a ludicrous proposition. That being said, I'll try to hit the highlights for you. There are 3 types of photoreceptive light cells in the eye: rods, cones, and photosensitive retinal ganglion cells. The last category doesn't have much to do with the sense of vision, but it's thought to have something to do with circadian rhythm or pupillary reflex. Whatever those things are. The rods are responsible for low light situations, and while they are quite sensitive, they also don't contribute much to the sense of color vision. Which is a long way of saying, let's talk about cones. The 6 to 7 million cones that live in an average human eyeball exist around the macula, an area near the retina in the center of the eye. There are 3 types of cone cells, and each one is sensitive to a particular area of that 300 nanometer visual spectrum. And back to chemistry. Here I've drawn a figure of intensity versus wavelength, so you can see where each of the 3 cones responds best. The long type cone responds best around 560 nanometers and is called the L cone. Go figure. This is the red cone. M-type cones, you guessed it, medium, respond best around 530 nanometers and are the green cone. Finally, the S-type cone, and I know this is going to blow your mind, but short, responds best at about 420 nanometers. Blaze it! This is the blue cone. And with this, you can see how the cones encompass the entire 400 to 700 nanometer range of visible light. In addition, and forgive the drawing, I've made a very purposeful effort to show how the spectral ranges of each cone overlaps the other ones, and this is critical. The brain uses an opposite process to figure out what color is being seen. Restated, the brain uses the intensities, and therefore wavelengths, from each of the cones to determine which wavelengths don't overlap with one another, and that fine-tunes the color that actually is being detected. And that's what happens when an average human eye sees color. What happens when something goes a little bit haywire, though? We'll talk about that and more in episode 2, but that's for then and this is for now. See you next time.
Can you sing with all the voices of the mountain? Can you paint with all the colors of the wind?